Okay, well, we'll go ahead and get started. I am working on the study guide for Chapter 3. I looked at the problems in the back of the text for Chapter 3, and they were kind of not that great as far as I was concerned. They involved math and calculating things, and that didn't look particularly fun. So I'm writing a study guide where I still want you to read the chapter because it's going to be very important, but then I'm going to give you a list of papers from the literature about non-covalent interactions. And I just want you to scan through each one, identify the major point, and write a 1 to 3 sentence blurb about what that major point is in the context of what we've been studying. And I'll post that on Learning Suite a little bit later today. Just as a reminder, many of you I think have had the chemistry writing class already, but let me give you a sense for how best to read the literature in this way. Start with the title, the abstract, and then the figures. Maybe look at the tables. Don't worry about the experimental section unless you really care and want to reproduce the work and want to understand it in depth. If you do that, you may get a general sense for the gist of the paper, and then you can read things out of the introduction and results and discussion sections as needed. I used to get bogged down in trying to digest each and every word of a paper, and if you try to do that, you're never going to read one of those papers, let alone 5 or more. So that's sort of my approach to getting a sense for what a paper has done, and hopefully that'll make the assignment a little bit more manageable. And once I post it, you can let me know if you have any questions. So we were talking about non-covalent interactions. A lot of these, most of them are electrostatic in nature. That is, they're related to interactions between charges, either full charges or dipoles or induced dipoles. Each of them has a distance dependence, and they get more complicated the more you start to involve dipoles and induced dipoles, the more complicated the distance dependencies get. We talked about Coulomb's law listed here, which tells you that the energy of an interaction between two charges that are fixed in space is related to the charge on one, the charge on the other. If they're opposite, the interaction will provide negative energy, which is favorable, and it will depend on the distance. This R here represents distance, and then we talked about dielectric constant, where higher numbers here, more polar media, higher numbers for the dielectric constant, increase the denominator, thereby decreasing the overall energy. Similarly, higher numbers for radius decrease the energy. That makes sense. The further apart charges are, the less they see each other. So sometimes people will say that the energy expression for Coulomb's law has a 1 over R relationship, or they'll say falls off as 1 over R, sort of a weird way of saying that, but what they mean is that the energy is inversely proportional to the distance between the two charges. All right, so talking about salt bridges so your text talks about ion pairs, which are common in organic solvent. This is where the two ions are still fairly closely associated, and this is how a lot of molecules that would in water dissociate into ions would stay in an ion pair in organic solvent. But when you put the ions in water with a large dielectric constant, the energy of interaction is not as strong and can be replaced with interactions between water and the dipoles on water and the individual ions. And that's how salts dissolve. An important kind of electrostatic interaction that plays a role in biology is this concept of a salt bridge. This is not a salt bridge in the electrochemistry sense. I think they used to talk about making some kind of galvanic electrochemistry cell, and then you'd have this tube, a U-tube filled with, not U-tube like YouTube we watch, but a tube shaped like a U, filled with salt water that you turn over, and it would provide a path for ions to go from one cell to the other. That's not the salt bridge that we're talking about. A salt bridge in proteins consists of, or nucleic acids, consists of two oppositely charged functional groups at some distance from each other that it seems reasonable that there might be an interaction. And a lot of people are interested in knowing how much each of these individual reactions is worth. It turns out that if you have a protein like this blob that we've drawn, and we'll assume that this is what we've drawn as the native state, and then it's in equilibrium with some denatured state, or we'll call it a denatured ensemble because it's likely a bunch of disordered conformations. We don't know what the denatured ensemble necessarily looks like. I'm going to draw a squiggly line to suggest that it's unfolded, but what that state looks like has been somewhat controversial. But there's some free energy associated with going from the unfolded to the folded conformation. And for a lot of proteins, that energy is on the order of 5 to maybe 20 kilocalories per mole. And there are a number of things that drive protein folding. The most important one is called the hydrophobic effect, which we're not going to get to until the end of chapter 3. And then there are these non-covalent interactions, or NCIs. And it's interesting, people want to know how much these individual non-covalent interactions are worth so they can understand protein folding and maybe design proteins with new structures and novel functions. One thing before we even talk about salt bridges that I want to make very clear is that whenever you're thinking about the impact of a non-covalent interaction, and you might want to ask how strong, there is no absolute scale for strength, okay, for non-covalent interaction strength. It's always how strong relative to what, okay? So in Coulomb's law, this energy expression is for the energy relative to infinitely separated ions where R equals infinity. And at R equals infinity, energy goes to 0. That's your reference point. In terms of protein folding, if you wanted to know, is having these two charges close to each other, how much is that worth? You have to consider context, okay? What do you mean? Do you mean for what would it be like if you had the folded protein and you just clipped away those charges? Or do you mean of the free energy that is from the unfolded state to the folded state, how much do we owe to having these two charges together? And the answer gets kind of complicated, in part because even though you may think these two charges are close to each other, if you're going to say they contribute to the stability of the native state relative to the unfolded state, you have to remember that the unfolded state is surrounded by water molecules too. And in this case, those charges, we already know that salts dissolve in water, right? Because water is good at orienting itself around both positive and negative charges, various dipoles. So the real question is this arrangement of salt bridges, is this arrangement having two charges close to each other better or worse than having two charges far away from each other but interacting with water? Okay? If you forget the water, you're going to be tempted to conclude, oh yes, relative to being infinitely far apart, that's a very strong interaction. But that's not our relevant reference point for protein folding, because the two charges, though they may be far apart, are not in a vacuum. They are surrounded by water. Okay, so you have to remember that. And even in the folded state, you have other water molecules that can surround the ions, and so depending on how close they are, there may not be a very tight interaction. Because of this, it is estimated that salt bridges on the surface of a protein are generally weak, on the order of 0 to, I forget what the number is, on the order from 0 to 2 kilocalories per mole. So surface salt bridges, you can think of 0 to 2 kilocalories per mole. One question, and it's controversial again because of sort of what the correct reference state is, but this idea that if you bury a salt bridge, you put two things inside the protein, they're in a medium that is not polar. You can think of protein interior as having an effective, nonpolar protein interior as having an effective dielectric constant of, somewhere between 4 and 37. So not like water, but not quite like hexane, somewhere intermediate. And a lower dielectric constant than water should strengthen the interaction between those two ions. And that's true, but you could ask the question, is it better to have a buried ion pair in a protein or just have nonpolar residues there in place? And if you do ask that question, sometimes even though the interaction between those two charges is strong, it's not better than it would have been to just have nonpolar residues there in the first place. So what I'm saying is that the salt bridge strength in many biological contexts is a weak effect, 0 to 3 kilocalories per mole, and it is context dependent. You can predict that from the Coulomb law with the radius and the dielectric constant, but when you're talking about biology, you must also remember that water is around, and so you have to account for the impact of having water nearby when the ions are not together versus nearby when the ions are together. Questions? You want to talk about that at all? Okay, so what I hope to communicate to you is even though these individual non-covalent interactions alone are very weak, there are many of them in a protein, and any individual one, if any individual one is worth 0 to 2 or 3 kilocalories per mole, that's not that far from the overall stability of the protein to begin with. And so you can make substantial changes to protein folding free energies. You can make, and I guess this number should be negative because protein folding is usually downhill. All right. There's a couple of things from my lab's research that I thought I would take a moment to tell you about. There are two of them that I could tell you about, and maybe I will just do some, but not all. So let's see. I don't really want to talk about this one, but it has a really funny graphical abstract that I sort of want to talk about. So in my lab, one of the things we're interested in is what happens to protein side chains when you modify them with unnatural groups. And for various reasons, people for a long time have been attaching a polyethylene glycol chain to protein surfaces, in most cases with the goal of making better protein drugs that last longer in serum and don't get degraded by proteases or recognized by antibodies. But we've been interested in my lab on understanding what that side chain modification does to protein stability and to the non-covalent interactions near the attachment site. And our hypothesis has been that PEG locally desolvates the protein surface, thereby making nearby non-covalent interactions stronger. There's some evidence for this, though we don't understand the effect completely. And I think my grad student Chung Xiao came up with this idea And then I drew the little cowboy in in Notability, and we had to trace the whip from somewhere because we couldn't figure it out. And then I think maybe Steve Draper came up with the idea to have the little water molecules going out. Do you remember? I can remember being in the office.
Did the faces,
Okay.
water molecules.
Yeah.
I think Steve thought of the whip, but then he just put in the whip as right, and then
Yep.
spent like 20 minutes tracing it.
That's right. So this has been the most creative graphical abstract we've ever done, and I don't know that I'll ever do it again. I think we've been all business ever since then. But
See if I can record you.
what the point of the study was to take. Point of the study was to measure the strength of various salt bridges on a number of different protein model systems, and to do it with and without a nearby peg. So in the figure I'm showing you here, panel B you can sort of ignore. This just shows structures of how we've attached peg to protein side chains. You've got without peg and with peg, a couple of different options. In this panel shows you the locations of places where we've investigated salt bridge strength with and without a nearby peg. So as an example, in this particular protein, which is a alpha helical coil, you have a positively charged lysine and a negatively charged glutamate, and they're on adjacent turns of an alpha helix, which sort of puts them within 3 or 4 angstroms of each other, and that distance is consistent with a salt bridge. This is crystal structure data, which doesn't necessarily tell you what's going on in solution. The way you measure the salt bridge strength conventionally is to mutate each side chain individually to a non-charged surrogate, and you can make arguments as to which choice is the best. In many cases, alanine, which is just a methyl group, is chosen as the best side chain. It's comfortable in many environments, and it's especially in alpha helices. So it's thought that truncating from a charged side chain to alanine is sort of as conservatively neutral as you can get, changing only the fact that the side chain is charged. And that's an assumption that people deal with. But you mutate each one individually to alanine, then you mutate both of them. Compare that to the stability of the starting compound, and you can measure the strength of the salt bridge between these two groups. Then you do the same experiment with a PEG side chain at an adjacent site, and you say, how much did the salt bridge strength change with and without PEG? In this case, almost no change, got a little worse. Over here, it got modestly not really even better. In fact, we only found 2 cases where we observed substantial stabilization, and it was with this particular protein where having PEG in an adjacent position strengthened the salt bridge by 0.7 kcals per mole, and again by about 0.4. And we summarized the data in this graph, which basically shows you salt bridge strength without PEG, salt bridge strength with PEG. Slope of the line is 1. Most of the time it doesn't make a difference except for these 2 variants, and we don't totally understand why. Though a crystallographic experiment revealed that if you zoom in on the site, if you get a crystal of the protein without PEG and the protein with PEG and zoom in on the site where the salt bridge is. So here is I want the laser pointer. Where is the laser pointer? Okay. Here is a glutamate, here is an arginine. Attaching PEG at this position was stabilizing by about 0.7 kilocalories per mole. Here's the protein that's PEGylated. Here's the crystal structure. We were not able to resolve the electron density for the PEG side chain in any ordered way. But here, this is the, what is this called, Kim? Electron density map from the crystal structure. Showing electron density that is not accounted for by our model. And even though the PEG side chain is disordered, we think it is occupying this space near the other salt bridges, near this salt bridge, that otherwise water would occupy. And that may be one of the reasons why the salt bridge is getting stronger. Did I miss anything, Kim? No? The other thing is actually Kim's project in my lab, so we'll just keep going, because why not? Unless you'd rather not. You can tell me if you'd rather not, but I don't care. Sorry. Yeah, so did I get everything there? All right, well, anyway. So in this project, we were looking at the surface of a,
Hey, Josh.
yeah.
Dakota has a question.
Oh, sorry, Dakota, go ahead.
Is PEG some new kind of amino acid, or is it a substituted
Um,
amino acid?
PEG is just, we're taking an existing amino acid here, asparagine, and then we're modifying the amide nitrogen with this polymer.
With polyethylene glycol?
Yeah. Yep. All right.
Is that ever found in natural protein?
No, but a lot of people are doing it now. The chemistry is pretty straightforward. In this project, we were looking at a three helix bundle, trimer, and we identified two side chains on the surface that are 2 helix turns apart. This distance, I think, is about 16 angstroms, which ought to be too far for them to interact via a salt bridge. And if you look in the crystal structure, they're nowhere close to each other. But we wanted to know, does the identity of the side chain in between these two groups impact their ability to see each other? And the story is kind of complicated. It turns out that when you have small side chains and relatively polar, small side chains, that the two charges don't see each other very well. So when serine occupies this central position, the salt bridge, or rather the putative interaction between these two amino acids, glutamate 10 and lysine 18, is unfavorable. But we found that with larger side chains like tryptophan or tyrosine, there was a measurable interaction, or rather synergy, that's not explained by either amino acid on its own, that occurred between glutamate 10 and lysine 18, but only when tryptophan was there, and that was worth about 0.7 kcal per mole. You could change it to tyrosine, and that was, and the effect was about the same. We learned that having a hydrogen bond donor on the edge of the ring was important, because when we replaced it with the methyl group, suddenly those two side chains didn't interact in a favorable way anymore. We tried to rationalize what was going on, and it turns out that our data sort of fell into two different categories. So on the y-axis is strength of a salt bridge, and on the x-axis is basically the hydrophobicity, or the log p, which we talked about last time, of the side chain at that central position. And in blue up here are side chains that generally lack a hydrogen bond donor on the edge of the ring, like methyltryptophan or methyltyrosine or phenylalanine, cyclohexylalanine, any of these others. And then in orange are variants that do have a hydrogen bond donor at some point out on the edge of the side chain. And in both groups, we observed that less polar side chains give you more favorable interactions between the two orange amino acids. So there's two different things going on that are helping that interaction occur. You need not only a nonpolar side, you need some amount of nonpolar surface area in between the two, and then you also need a hydrogen bond donor for the maximal effect. The story gets even more complicated than that, though, because we later discovered that everything we just learned depended on the position of another charged residue on the other side of one of them. And so what we're learning is that interactions between charged groups on the protein surface are actually pretty complicated, but can contribute a lot to protein stability. And so we're trying to understand what all of the context-dependent rules are. You could imagine that the side chain in between these two groups is, you need some nonpolar surface area. Perhaps that is excluding water from the space between these two charges. It's also possible that the side chain here, if it's big enough, can interact favorably with lysine, and that frees this glutamate up to form a salt bridge with this lysine. We don't know, and studies are ongoing, but did I miss anything, Kim?
Sounds about right.
What's the big punchline? What would you say?
What's the big punchline?
Yeah.
It's complicated.
Okay, non-covalent interactions in protein structure are complicated. Yep. But just because something is complicated doesn't mean you shouldn't study it, I guess, is
Asking questions.
keep asking questions. All right. Another type of interaction is ion-dipole interaction. This is the kind of interaction of, for example, water solvating an ion. If you think of a dipole as being a linear distribution of charge, positive on one end, negative on the other, and if you imagine fixing that dipole and then having some other charge nearby, and the distance between that charge and the midpoint of the dipole is r, and then the angle that that line forms with the dipole itself is theta, then I'm not going to ask you to memorize this, but I just want to demonstrate that there's still an energy relationship, but because the dipole is not a point charge, it depends on the angle of the dipole, orientation of the dipole relative to the charge. The denominator still has dielectric constant in it, only this time it's a radius squared dependence. So dipole ion interactions fall off faster than ion-ion interactions. They fall off as 1 over r squared versus 1 over r. Okay? Dipole-dipole interactions, I could write the math, but I don't think I will. I will just say that dipole-dipole interactions fall off as 1 over r to the 3. So these are getting ever more short range, right? Yeah.
Yeah, we worked out the math for this in Physics 220 somewhere, I think. And
Okay.
It involves some kind of average or Taylor series or something like that.
Yeah, the term for the angle between the two dipoles is 3 times cosine squared of theta minus 1, and you have to assume that the two dipoles are coplanar in the same plane and parallel to each other. Other than that, I think it gets even more complicated. But as you start to move from full charges to smaller charge increments, the interactions get shorter and shorter in range. Okay? A specific kind of dipole-dipole interaction, which is often discussed as its own separate thing, is hydrogen bonding. And this is a favorite and important topic, and it's the one with which you have the most experience because we live in a world of water, and we ourselves are mostly water. That I'm going to get myself in trouble here by quoting an early episode of Star Trek, The Next Generation. No, nobody recognizes it. Okay, it's on Netflix, people, and it was on in the 90s, so it's not that old. You might consider looking it up. At one point, Patrick Stewart's Captain Picard is encountering a superior life form, which refers to humans as filthy bags of mostly water. Okay. In any case, a hydrogen bond is described as an interaction between a polar bond between a donor and a hydrogen, and the donor is usually electronegative, and the hydrogen usually has a partial positive charge on it. and then some acceptor atom that's also electronegative and has a partial negative charge on it. And this is primarily an electrostatic interaction. So the orientation of the groups relative to each other isn't that important. In other words, you can move this acceptor around. The acceptor and the donor and the hydrogen don't have to be collinear. They can be offset by some amount, and the interaction is still okay. Though, to a first approximation, you can sort of get a feel for what optimal hydrogen bonding geometry might be by considering where the lone pairs are on the acceptor. So if we think of some hydrogen bond donor, we won't say whatever the, sorry, we'll just keep the DN. And if we think about how that would hydrogen bond with an amino group, remember the geometry of ammonia that we learned about at the beginning in chapter 1, where we said that group orbitals place the lone pair in a sigma out kind of orbital, which is pointing away from the other carbon-hydrogen bonds. You would predict the most effective geometry would be the one where those lone pairs are lined up directly with the hydrogen. In contrast, if you're let's see, if your hydrogen bond acceptor is a carbonyl, in many cases, being in some medium that can hydrogen bond allows the sp or the S-rich lone pair to be able to mix with the P-rich lone pair, and you end up with rabbit ears like lone pair orbitals. And in many cases then, if you have the hydrogen bond donor here, and if you were to measure sort of an angle between the line, the angle between the line that starts with the carbon and ends with the oxygen, and then the line of the hydrogen bond, it tends to be about the right geometry for what you would expect. Yes?
Guys, remembering from structural biochem, we talked about parallel versus anti-parallel data sheets.
Uh-huh.
And how the way that the hydrogen bonds line up is kind of like the determining factor of their strength.
Uh-huh.
And how I don't even remember which one. I think in anti-parallel they line up at 180, and in parallel they line up offset.
Yeah, very good. So let's look at beta sheets, parallel vs anti-parallel. Fight!
It's been good for me, so.
All right. What? This is a cruddy webpage. This is also a somewhat cruddy webpage, but it'll do. Yeah, in the anti-parallel beta sheet, you can get almost sort of linear arrangement. And again, you might think, what the heck is going on? Because I thought now we were saying the lone pairs are sort of in sp2 hybridized rabbit ears, only when context demands it. If you want a linear arrangement, you're going to have one lone pair in an s-rich orbital pointing directly at the hydrogen, and then the other in a p-rich orbital orthogonal to that. I'm going to put a paper on your study guide where the Rains group actually talks about the possibility of a hydrogen bond between a p orbital here and that proton, or rather that one. So an intra-residue hydrogen bond, kind of weird. Yeah, and then in the parallel beta sheet, it's offset, and so then it would be more sort of your rabbit ear type orbital. As to the relative energies between the two, I don't know, actually. Though people...
All anti-parallel being more stable, and they rationalize it by the strength of the hydrogen bond being better when it's linear.
Yeah, some of the early Gelman literature was comparing anti-parallel and parallel beta sheet model systems, so there might be some information there. I don't know. What your text says is that hydrogen bond geometry generally, I mean, there are effects, but it generally doesn't make that big of a difference. Okay, so what do we want to say about hydrogen bonding before we beat it to death? Strengths of hydrogen bonding tend to are also context dependent, as you might expect. The very strong ones tend to be in the sort of 15 to 40 kilocalories per mole. So that's approaching weak covalent bond character. The sort of moderate ones are 5 to 10 kilocalories per mole. The weak ones are 0 to 4 kilocalories per mole. As before, the strength of a hydrogen bond depends on what your comparison state is. As an example, my PhD advisor in his early work just after he started his job at Wisconsin, and this was the stuff that got him tenure, was studying hydrogen bonding via NMR and IR in very small model systems. So they made this diamide. Let's see, I'm going to draw it wrong probably. So that's a bond coming out at you and going back into the page. We're looking at it directly on its side. That's what you get if you tip a methyl group sort of over. Oops. Very carefully controlled this molecule so that there was only one possible hydrogen bonding interaction. And they measured its stability, I believe, with variable temperature NMR. And they found that at least in terms of enthalpy, which is strength of the interaction, this hydrogen bond was worth somewhere between 1.4 and 1.6 kilocalories per mole. But the solvent was dichloromethane, which is, of course, a somewhat modestly polar but aprotic solvent. What do you think we would observe if we change the solvent to water? Right? Why? It's just as easy to hydrogen bond with water, right? Probably the strength of the hydrogen bonds between this molecule, let's see, 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. I'm not sure if I'm drawing the right number of carbons. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Good. The strength of hydrogen bonds between these groups and water is probably just as strong as it is in the sort of folded up structure. And also, there's an entropy penalty. Entropy goes down by around 6.8 to minus 8.3. I hate this, entropy units, whatever the heck that means. I think that's calories per mole Kelvin, but anyway. And there's a lot more ways for water to hydrogen bond with these groups than for them to hydrogen bond with each other. So there's an entropy argument there as well. Go ahead, Isabella.
How would you measure the entropy of that?
How would you measure the entropy of that? That is a fantastic question. And Usually that is by Hoff Analysis. So what you do is you remember that delta G equals minus RT ln K. Then you remember delta G equals minus H delta H minus T delta S. And you divide both sides by RT. And then you plot log of the equilibrium constant versus temperature. And your slope should then equal delta H over R. And your intercept should equal minus delta S over R, I think. Your text describes how to do it, and I may have butchered it, but that's one way you can do it, is you measure the equilibrium constant as a function of temperature. Presumably they did this, I can't remember, probably by variable temperature NMR. If the two conformations are in equilibrium and the equilibrium is slow on the NMR timescale, you'll see chemical shifts for both. And then you can integrate them for population, how much is here, how much is there, calculate an equilibrium constant, and then just crank up the temperature and watch it change. All right, so that's a key point. Hydrogen bonds in water, how strong are they? Well, relative to what? Relative to the alternative of hydrogen bonding with water? Probably not that strong. But the strength is context dependent, and as you might expect, if you have hydrogen bonds that are in an environment where they are sheltered from water, sometimes the strength can be substantial. So I'm going to show you a few, because I like proteins and hydrogen bonds, I'm going to show you a few examples of hydrogen bonding. And don't pay very much attention to the notes I've already scribbled on this. In fact, what we'll do is I'll just copy this and then we'll paste it there, and then I can erase stuff. All right, sorry, large. So this is the alpha helix, classic hydrogen bond structure. You have a carbonyl oxygen at, we'll call it the I position because that's where I am starting to count. Thank you. Then you've got I plus 1, I plus 2, I plus 3. You get to the I plus 4 position, and you have an amide nitrogen that belongs to that I plus 4 residue, nitrogen, alpha carbon, carbonyl carbon, and they're engaged in a hydrogen bond. And the alpha helix consists of a regular array of these hydrogen bonds, every amino acid. Probably based on the geometry, it's probably the S-rich lone pair on the carbonyl oxygen that's interacting with the electron poor hydrogen here. So you would expect, what do you expect on folding from an alpha helix? What should the change in enthalpy be? Well, relative to being unfolded, the carbonyl oxygen and the amide nitrogen are probably hydrogen bound whether folded or not, right? Either to water or to each other. So that's probably maybe not a huge amount of enthalpy change. However, in the helix, one of the things we haven't talked about is you've got these side chains that jut out off of every alpha carbon, and they can somewhat protect the inside of the helix from water. And so it's possible that the helix hydrogen bonds are a bit stronger than the hydrogen bonds with water. All right? But if you think of an unfolded protein going to something that's helical, what do you think entropy is going to be? You've got to restrict, right? It should be unfavorable. You've got to restrict the freedom of these amino acids. And so helix folding has been analyzed, but they think it's a process whereby the formation of the first few turns is very slow because the payoff you get in terms of enthalpy isn't quite enough to compensate for the restricted degrees of freedom that you get by folding into the helix. But that penalty or entropy starts to go down for each additional amino acid once you already have a few there, whereas the additional benefit in terms of enthalpy for hydrogen bonding continues to go up each time you add another amino acid incrementally. So it's thought that once you get beyond a certain length, you've overcome the entropic barrier to folding the helix, and now forming the hydrogen bonds is good enough to allow you to become helical. So there's this concept of helix nucleation, and a lot of work has been done to try to say, what can we do to the end of a helix to make it nucleate better? Go ahead, Kim.
Do they not take into account water being freed up at all in those situations?
Great. That's fantastic. I didn't mention that, but yeah, Kim's exactly right. You are going to free water, which, you know, those waters looked sad in that diagram, but actually they're very happy because now they have lots of entropy. So yeah, you're freeing up water by forming those hydrogen bonds. Yeah, there's more we could say about the alpha helix, but that's a classic example of hydrogen bonding. Any questions about that? Anything you want to talk about there? The whole strategy of helix stapling is related to that some people are working on in my lab and in the Michaelis lab. The whole concept of helix stapling is just related to paying for some of the nucleation cost to get the helix going. It's also a reason why you don't form alpha helices with very, very short peptides. You need to have 20 to 30 residues to really get one going. And even then, helices usually form after something that's called hydrophobic collapse, which we'll talk about another time. Let's see, you've seen hydrogen bonds in beta sheets. It's interesting to ask the question, what is one of these hydrogen bonds worth? And early on, people started to investigate the contribution of any given hydrogen bond by what's called amide to ester mutagenesis. That is, think of replacing one of these NH bonds with the corresponding ester. And you'd think, oh, that's fantastic. You got rid of the hydrogen bond and that's all you did. Well, amide to ester does a couple of things. First, the hydrogen bond is gone. Second, you've got a potential repulsive interaction between ester oxygen and the downstream carbonyl oxygen. Now they're going to repel each other, delta minus versus delta minus. And then finally, not only did you change that hydrogen bond, but esters are weaker H bond donors than amides are. Again, because of the electronegativity of oxygen. So you've changed a few things, but if you do the experiment carefully enough, you can sort of tease out how much a hydrogen bond is worth. The work that was done here early on in the late 90s when I graduated from high school, was done by Peter Schultz, who then went on to do a ton of stuff with incorporation of unnatural amino acids into proteins. But they found that in general, a hydrogen bond was worth about 1.6 kcals per mole. About half of that was due to the weaker hydrogen bond here, or the absence of a hydrogen bond here, and the other half was due to a weaker hydrogen bond with that carbonyl. People have continued to do amide to ester mutagenesis to evaluate hydrogen bond strength. Another paper I'll have you read comes from the Kelly lab, where they looked at how much a hydrogen bond is worth in the core of a protein versus elsewhere, and they found some interesting things. So I guess we're done. I intend to take exactly as much amount of time as I want on chapter 3, and we'll just neglect other things. So I think probably 2 more lectures on chapter 3. We'll eventually get to reactions, but anyway, so have a good Wednesday. We'll see you later.